Meiosis. In this video, we will be discussing this specialized cell division process, and it's important to understand the difference between this division process and the division process we talked about in the previous video, which is called mitosis. So that is a major concept that you're going to want to have down after reviewing meiosis and mitosis, the major differences between these two cell division processes. And with meiosis, this is again a specialized division process. It only occurs in eukaryotic organisms, and it only occurs in the reproductive organs of those eukaryotic organisms. It is again a specialized division process to produce cells called gametes, egg or sperm. If you are a lady, you will produce eggs. If you are a gentleman, you will produce sperm. The gametes are oftentimes called sex cells, and they are the product, or they are the major cells involved in sexual reproduction, which we will talk a little bit about in this video as well, as far as how those cells come into play in the process. Now, the type of cells we talked about when we looked at mitosis, that cell division process, those are somatic cells. Somatic cells are body cells. Any cells other than the gametes. So liver cells, brain cells are somatic cells, cardiac cells, muscle cells, those are all body cells, also called somatic cells. So you're going to want to know the difference between those two. And then the final concept here we want to talk about is what is a karyotype, and then how we can detect certain chromosomal abnormalities using these devices, these karyotypes. Okay, so let's go ahead and jump right into it here with the somatic as opposed to the gametes. So somatic cells, they have two sets of chromosomes, two sets. They have one set came from one parent, and the other set has come from the other parent. And we call these diploid cells, di for two, and they're represented by this number 2n. So the n is the number of chromosomes in one set. So for instance, in humans, we get 23 chromosomes from our mother. The eggs have 23. The sperm have 23. So the number in one set would be 23 in one set. So 2 times 23 is 46. Our diploid number is 46. Every single one of your body cells has 46 chromosomes. Every one of those chromosomes has a pair. So when you get one of those chromosomes from your mom, you get the same type of chromosome from your dad, although they might have slight differences in them. And we'll talk more about that when we get to the genetics chapters. Now looking at gametes, again those are egg or sperm, they have one set of chromosomes. One set. And the reason for that is because the egg and the sperm combine during fertilization and produce that diploid organism, that first somatic cell of that new organism. So we'll take a look at the diagram, which does a really great job of pointing out these different types of cells or cell types. Now for the gametes, we call those haploid cells. They have one set of chromosomes. Like I said, we have 2 of each chromosome in each of our somatic cells, one from mom and one from dad. That's why we have 2 of each. And they have similar placement of the genes along the chromosome. They are a similar size. They have a similar placement of the centromere. And when we look at these pairs, we call them homologs, the homologous pair. Every chromosome has a match, a homolog. That'll be important when we talk about genetics and looking at genotypes and such. Okay. So here we have a sexual life cycle shown here in this diagram. So the process of meiosis, egg and sperm combine. So we have egg produced by the female in her gonads, her ovaries, and sperm are produced by the male in his gonads, or the testes, and they produce haploid cells. these cells that have just half the number of chromosomes that your normal body cells would have. And the egg and the sperm combine to produce that first diploid cell. So when they combine, that process is called fertilization. And that cell, that diploid cell, then divides by mitosis and develops into the organism, either a male or a female. So we see both the diploid and then the haploid cells in this human life cycle. All right, so as far as the comparison between mitosis and meiosis, what I really want you to focus on are really the end results of the division process and what the major purpose is of the division process. So mitosis is shown here on the left. So in mitosis, again, we're just trying to make more of the same type of cell. So the types of cells we're producing at the end are identical, because we're just trying to replace cells that are damaged perhaps, or just grow and develop, make more of the same type of cell. So it's really, you're taking one diploid cell and you're making two identical diploid cells at the end of the process. That's important. Two diploid cells, identical. So for instance, in our cells, we have 46 chromosomes. So we start with a cell, a body cell, a somatic cell with 46 chromosomes. And at the end of the process, we'll have two cells, each of them having 46 chromosomes. Now, meiosis, that occurs, again, only in the reproductive organs. It's a specialized division process. So you start, again, with a diploid cell, just like you do in mitosis. But these are special diploid cells. These cells are destined to undergo this division process. And they undergo this division process, and again, I'm not really concerned with you knowing the specifics of the division process. I want you to focus on that end result. So from one diploid cell, it's a special diploid cell, again, in the ovary or in the testes, we're going to produce four haploid cells. So let's say we start with a 46 chromosome cell in the gonad at the beginning of meiosis. At the end of meiosis, we will have 4 cells, each of which will have 23 chromosomes, half the number of that original cell. That's really what I want you to focus on. This is a great diagram for focusing on that. All right. So on to a karyotype, and a karyotype is really just an ordered arrangement of a cell's chromosomes from largest to smallest. And at the very end, you'll have the sex chromosomes, the X and the Y here. So this individual is a male. If you saw an X and an X, you'd have a female. Now, you might notice here that the X is quite a bit larger than the Y in males, as we can see here. So there is a process that occurs to accommodate for this in the female. We aren't going to really discuss the specifics of that, although the process is discussed in one of the videos for chapter 10, I believe, chapter 9 or 10, if you are interested in the process. But for our purposes, for this course, we're just really interested in you understanding the concept of 2 X's is a female and an X and a Y is a male. And there are certain abnormalities we can detect, chromosomal abnormalities we can detect from a karyotype. For instance, trisomy, 3 copies of chromosome 21, as you can see here, will result in Down syndrome. So it's a whole series or set of characteristics and behaviors in this this individual that we see as a result of just having an extra chromosome. All right, and then there are some other chromosomal abnormalities. But this diagram or this chart really shows you that as maternal age goes up, so does the chance of having a child with Down syndrome. So oftentimes, not always, but a lot of times, a mother that is older, maybe her last child or a surprise child, upwards of 35 to 45 years old, more often that child is likely to have Down syndrome, because as the woman ages, so do her ovaries and so do her eggs, and the division process becomes less accurate and a lot of times the eggs will result in having too many or too few chromosomes. Now, other abnormalities, a lot of times you'll have abnormalities in the sex chromosomes. So, for instance, I have two shown here. One is called Klinefelters. We'll have two X's and a Y. The person is male, physically male, but the testes or the reproductive structures are undeveloped, and they actually have some secondary sex characteristics of females like breast development, and they won't really grow facial hair quite as well. And also, too, we have another condition of the sex chromosomes, Turner syndrome, where this is a female. She just has one X, and that's it. There's no Y, there's no second X. And similarly, the ovaries or the reproductive structures are really underdeveloped. Problems with the heart, webbing of the skin around the neck is characteristic. So you have, again, secondary sex characteristic abnormalities, and then just problem with reproduction in these individuals. So that is that with our meiosis chapter and looking at chromosome abnormalities and karyotypes. If you have any questions or need any assistance, please do not hesitate to contact me. And I hope this has helped you better understand this process.